So hello everyone joining us today or joining us from home online. We are from Team Tox, an organization in the toxicology department at Oregon State University. Today we'll be walking you through a science experiment based on yeast dose response. And all of that might sound like nonsense right now, but hopefully by the end of the activity, you'll understand what all those words mean. So the very first thing that I'm going to do before we even introduce ourselves is invite you to open up your toxic, non-toxic sack, or gather your supplies if you're doing this just based on the worksheet. And if you're using hand warmers in this activity, if you can't plug in a heat pad, then I would invite you to open those up right now so that they can start heating up, because they do take a second. If you have any questions about that, you can put them in the chat. And otherwise, I will start introducing ourselves. So I am Emily. I am a PhD student in the toxicology department at Oregon State University. And right now I am studying firefighter health and how chemicals that firefighters are exposed to can impact their health. Christian is joining me today to help present this activity, and I'll let him introduce himself.
Hey, Emily, thanks for introducing me. As she said, my name is Christian. I'm from Redding, California, which is about 6 hours south of here. I'm a first-year PhD student in the TOX program, and I don't have a lab yet, but what that means is I get to go taste test a few different labs and see which one is the best fit. Are we ready to begin, Emily?
Yeah.
Great. You want to bring us to the next slide?
Yes.
So we're going to jump into starting up our experiment really fast and then go into the background information afterwards so that we give our yeast lots of time to grow. Next slide, please. Okay, so in your bag for toxic, non-toxic, there should have been 4 little plastic baggies. The very first thing we're going to do is label those, because we don't want to get lost with what's in what bag. So we need to label one bag control, the next bag low, and medium and high, and so forth. And oftentimes it's good to put little indicators for what we're going to do with each bag on those as well, so we don't get lost with that either. That's why we have 0 teaspoons, 1/4 teaspoon, 1/2 teaspoon, and 1 teaspoon underneath those control low, medium, and high labels, so that we know when we're adding salt just how much to add. So I'll give you guys a little bit of time to write your labels on there. They may look something like this. When you're, I can't, you can't see that, never mind. Okay, once you've got them labeled, next slide, please, Emily. We'll start adding our ingredients. The first thing to add is 2 teaspoons of yeast and 2 teaspoons of sugar to each bag. I like to start by doing all of one thing in all the bags and then all of the next thing in all the bags. So I would add 2 teaspoons of yeast to my control bag, and then 2 teaspoons of yeast to my low bag, and 2 teaspoons of yeast to my medium bag, and so on and so forth. And then once I had all the bags filled, then do the 2 teaspoons of sugar to each bag. So once you've filled your bags, they should look something like this, with a little bit of yeast and sugar action in there. You can shake it up a little bit so that it's more homogenized. That's just a fancy word for mixed together. And then we can start adding our salt. So I'll give you a few more seconds to add yeast and sugar, because that can take time. Something else to think about while you're adding your ingredients is that you want to get as close to 2 teaspoons in every single bag as possible, so that you have the same amount in every single bag, because we want these to be the same so far anyways. Okay, when we add the salt, our high-tech labeling is going to become very helpful for us because we'll see on our control bag that we're not supposed to add any salt at all. So we'll just toss that to the side for now. We'll get our low bag and we'll add 1/4 teaspoon of salt. And after we've added our 1/4 teaspoon of salt to our low bag, we'll add 1/2 teaspoon of salt to our medium bag and 1 teaspoon of salt to our high salt bag. If any of this, if I'm going too fast, the instructions are on your handout, and that should help you get oriented. Now, once we've added salt to each of our bags, except for our control bag, we're going to go ahead and add some warm water. We like to use warm water because yeast will grow faster in warm water than it will in cold water, but we can't use like boiling hot water because just like boiling hot water would hurt you or me, it will also hurt the yeast. So I'm going to go ahead and add 2 tablespoons to my control bag. And I'm going to mix it around a little bit, and I'm going to close the Ziploc on everything except for the very end. And then I'm going to try my very best to get as much of the air out of it as possible. If you have a flat surface, you can kind of like hold your bag against it and push the air out, but be careful not to let go of any water. And once you've gotten most of the air out, like this, look at that, it's a skinny bag, then you're gonna seal it all the way shut, and then you'll either wanna put it on your hot pad. If you have an electric pad, you wanna put it on the towel over your hot pad. If you have one of the chemical hand warmers, you wanna put it directly onto that. Or if you have just a bowl of hot water, like I have, you can just put it in the bowl. And now we'll add 2 tablespoons of warm water to each of our other bags and follow the same air exclusionary practices. Ooh, good question, Emily. I do not know. Emily Bonner, do you have a idea of what is a good setting for our hot pads?
I have not used the electric hot pads before. So I would say, I don't know, maybe low, but I haven't tried them before. The low would probably be most comparable to the hand warmers.
Okay. And if you put your hand on it and you can't really tell that it's warm, then you probably want to turn it up. But if you put your hand on it and it burns you, then probably turn it down a little bit. Once you've got your water and all your baggies, and they're all sealed tight, we're going to let those sit for about 30 minutes while we go over all of our background information and everything for the experiment. I'm finishing up my last bag as we speak.
I might need an extra second.
Uh-oh.
It took my water a second to heat up.
Thank you for that warning there, Emily. Appreciate it.
Yeah, of course. I will also be using the hand warmers, so I'll show you all how I'm setting those up.
Emily, are you doing multiple trials then, like a hand warmer trial and a hot water trial? Or are you just doing hand warmers?
Yeah, so I've done the hot water before, and that has worked well. I wanted to try the hand warmers to compare them, just to see what kind of height we might expect.
Look at that, I'm already getting bubbles. when you're ready, Emily, we'll move on. I'll take you as the representative sample of timing.
Thank you. I'm getting close.
Wow.
Okay, so I have my baggies. I'm gonna try to show how I'm setting them up on my sheet pad if I can. So I have my 4 little hand warmers set out. They're warmed up a bit at this point, and I'm putting each of my baggies sort of on top of them to help keep the yeast warm.
Nice.
And we'll see what happens.
I have all my baggies in a bowl of water that apparently you can't see the baggies through, but they're in there, I promise.
I will go to the next slide.
Thank you, Emily. We're going to talk just a little bit about our experimental setup. In particular, we're going to talk about our variables. Variables are anything that can change in an experiment, which means there's typically quite a few variables per an experiment. But there's only 3 kinds that we care about. And those are the independent variable, the dependent variable, and the control variables. The independent variable is what we purposely change in an experiment. So in this case, we purposely change the amount of salt that we have in each bag because we're trying to figure out what could salt do to yeast. The dependent variable is what we try to measure in the experiment. So in this case, well, I'm not going to say quite yet. Let's let you guys think about what is it that I could be measuring right now? Do you have it pictured in your mind what you're measuring? Okay, we're going to measure the volume of air in the bags by measuring the height of the bags. So that's going to be our dependent variable. And then control variables are anything that we keep the same in an experiment between trials so that we can tell what the independent variable is doing instead of like what something else could be doing on the side. So ask yourself, what are the things that we are keeping the same between each of the baggies? One of the control variables in this experiment is the sugar. We're using the same amount of sugar in each bag. Try to think of at least 2 other control variables in this experiment. Emily, do you have an idea of what one of the control variables might be?
Oops. They should all be the same temperature, right?
Yes, that is another well-controlled variable. Okay, now that we've gone over our variables a little bit, Emily is going to start taking us through some background on yeast respiration.
Thanks, Christian. All right, so this might seem unrelated, but I promise it will circle around to our yeast and what they are doing right now. So the first thing we're going to talk about is the fact that all living things need food. That's right. So there are different ways that living things can get food. So for example, if you have plants in your house, they're using photosynthesis. And this is where they will take in light from the sun usually, and then they will use that light to make sugars, which is their food, from carbon dioxide that's in the air and water. And they can use these sugars immediately, or they can store them for growth or later use. Animals or humans like us, we obtain food from eating plants or eating other animals. So if you eat a salad, you're eating those plants that have the sugars in them that they got from photosynthesis. Or you might eat a burger, and that animal that you're eating also has those valuable sugars that you need. Oops. Looks like there's an issue with my chat panel. Let me see if I can close that. Okay, so the next thing is being able to convert that food into energy that we can use. So the food that we take in actually goes through a series of chemical reactions. So what I mean by that is it is changed chemically. Things are broken down and added together and reformed. And so this can create new molecules that can support your growth or release energy. And in most animals and plants, oxygen from the air will react with sugars, which contain carbon, and those provide us with energy. In this process, we also produce carbon dioxide. So when you exhale, there's carbon dioxide coming out of your body. And this process is called cellular respiration. That's a big word, but really all it means is you're taking in this food, which is then converted into other molecules for you to use for energy, and you expel carbon dioxide. And our yeast are actually doing that right now. Okay. So I will let Christian talk about model organisms.
Okay, an organism is just any living thing. So I'm an organism, you're an organism, Emily's an organism, trees are organisms, dogs are organisms. Take a moment to think about what your favorite organism is right now. I think for me, my favorite organism is a giraffe. I saw in the chat that someone said ice cream was an organism. I don't know if that's true. I'm seeing shaking heads. I don't think that ice cream is an organism because ice cream's not alive. Model organisms are any organism that scientists study to help understand life. I'm now seeing that someone is offering cows as an organism instead of ice cream, and that would be a correct answer. Cows are alive, therefore they are organisms. So like I said, a model organism is any organism that we study to understand life a little bit better. Oftentimes they are used to help us understand ourselves, to understand humans better, or some other organism that is a little trickier to study. Usually model organisms are pretty easy to raise in a lab and have lots of babies and are not picky about what they eat. So as an example, if you wanted to know something about pandas and you thought you needed to study at least 1,000 pandas to understand it, that would be pretty hard. But if instead of raising pandas, you raised mice, it'd be pretty easy to raise that many mice and see what they did, and then try to understand what pandas might do similar to mice. Next slide, please. Here on this slide, we have some fun model organisms. So on the bottom left, there is a fruit fly. You may have heard of people studying fruit flies. Well, it's true. They do get studied. On the middle right, there's something with its arms out like this and little hairs on its arms. That's called a daphnia. And we use that to study lots of freshwater bugs. On the bottom right is a zebrafish. And those are little fish that normally live in India, but we've taken them into labs and we use them to study all kinds of aquatic fish and also humans. Zebrafish can help us understand a lot of things about humans too. I think the top middle is yeast, which is what we're studying right now. And then in the very middle, we have a group of cells. And sometimes instead of using whole organisms, we'll just use cell cultures or little tiny miniature organs. So that's kind of what you see in the middle there. And on the left, some really fancy people make things called organs on a chip. And I think that's what that thing on the left is supposed to be. But that's just another way to understand, to try to understand some living thing. Next slide, please. So we're using yeast as our model organism today. yeast works pretty well for us because it's pretty easy to grow in a bag. It's not too picky about what it's eating. We're just giving it water and sugar, and it's pretty easy to catch. I mean, we just kept it in a bag, so not so bad. They are little tiny microscopic funguses. And you may have looked at your yeast and thought, I can totally see that. What do you mean it's microscopic? Well, really what you were seeing was lots and lots of yeast. And those little pellets were not just a yeast. Those little pellets were like 1,000 yeasts in a pellet. So pretty small. Yeasts, like us, like to eat sugar, and they require water. And when you give yeasts water and sugar, they can make carbon dioxide and alcohol. So the carbon dioxide is actually what we're measuring in our bags as we speak. So if yeast are healthy, they will make CO2, and the bag will fill up with CO2. Next slide, please. So think about what yeast need to thrive from the last slide. They need sugar. They need water. They need, do they need salt? I'm seeing a shaking head. So we added salt, and we're going to see what that does to them. We're going to see if salt is toxic to the yeast. So if the yeast are feeling healthy, they'll make lots of CO2. So if the salt is toxic for the yeast, what will we expect the yeast to do? Will we expect it to make lots of CO2, or do we expect it to kind of peter out maybe? I'm expecting that if the salt is toxic for the yeast, it's going to peter out, not make a whole lot of CO2. But that's why we're doing an experiment. We won't know until we see. I think it's your turn now, Emily.
Thanks, Christian. That was great. So at the very beginning of this presentation, I said that Christian and I were from the toxicology department. And so now we'll talk a little bit about what that means. So Christian was just talking about salt and how it's maybe toxic to the yeast, and we're going to test that. And really, as toxicologists, all we do is we study toxins or poisons. So what is a poison or a toxin? Basically, it's anything that's harmful to living organisms, and it can cause illness or disease. So when we have something that might be toxic, we really need to consider how much of that chemical is ending up in an organism. Because if you have a lot of something that you're being exposed to, it could be more harmful than if you're just exposed to a little bit. So the amount of something that you are encountering, that is something that we call a dose. So that's just a measure of how much of a chemical you're being exposed to. So some examples here to think about are, if you've heard of arsenic, which is metal, is that toxic? Have you heard that? What about butter? You know, you hear that cholesterol and fats are not necessarily good for you, but is it toxic? What about water? You're told all the time, drink more water, I'm sure. My mom's solution to everything was always drink more water. You know, you're feeling tired, you're feeling sick. Did you drink enough water today? But can it be toxic? So an example is here, we have two people, one person's drinking 1 bottle of water, and then we have this person who's chugging a whole big jug of water. And actually, if you have too much water, if you were to drink like this jug of water, that wouldn't be good for you, and it would actually cause problems in your body. Fun fact, if you drink an enormous amount of water, like a lot of water, it can actually cause comas. So it's not to say that water is toxic, but it definitely depends on how much of something you are being exposed to. So how do we know if something is toxic or how much of something is harmful? We call this toxicity, and we can measure it based on the responses that we see in experiments. So if we have an organism and it is having increased bad responses, like if we're studying mice and a lot of them are getting sick when we expose them to a chemical, then we might think that it's toxic. We can also measure good responses. So say you have a bunch of mice, but there aren't as many mice being born as you would expect. Then you might also say that the chemical that they're being exposed to could be toxic. So what we're doing with our yeast right now, with the salt, is we're measuring a good response, which is the carbon dioxide is being produced and the yeast are growing. So if there is a decreased response or like a decreased amount of carbon dioxide, then we might say that the salt is toxic. So the reason that we test things and perform experiments on chemicals is to figure out if they're toxic or not. We don't know until we test them. And we want to make sure that we test them at different doses or different amounts. So like with our yeast, we're testing different amounts of salt because we don't know how much is too much either. So if we have a chemical and we don't know if it's toxic or how much of it is toxic, but we want to find out for humans, we don't want to test that out in humans because we don't know what impact that's going to have on them, right? If we don't know if something is toxic and we want humans to test it, that could end up making them sick, which is not good. We don't want that. So we might use a model organism like Christian was talking about. And we don't know how much. So we want to test different doses of the chemical. So like I said, in this experiment, the response that we're measuring is the height of the bags, which should show us how much cellular respiration is happening, which is a healthy response. And actually that's something that is shared by both the yeast and us. So if we see a decrease in carbon dioxide, we might expect that those yeast are not as healthy, and that could be some indication of the chemical in humans. So with that, let's get back to our yeast.
Okay, before we go and get real deep into analysis, we're going to want to think of a hypothesis. So a hypothesis is a prediction of what we expect to see in the experiment. A good way to make a hypothesis is to ask a question about your experiment and then make a pretend answer to it. So for instance, we might ask, if salt is toxic to yeast, do we expect to see less CO2 in the bag? Well, let me start over. If salt is toxic to yeast, would we expect to see a higher dose of salt to increase or decrease the amount of CO2 in the bag? Then we can try to answer that question with a statement. So if we increase the dose of salt that the yeast are exposed to, then blank. And we get to fill in the blank with what we think will happen to our response variable, which is the volume of CO2 in the bag. So I'm going to write down, if we increase the dose of salt that the yeast are exposed to, then the volume of the CO2 in the bag will also decrease. No, will decrease, not also decrease. Next slide, please, Emily. A good way to help us understand the answer to our hypothesis sometimes is called a dose response curve. A dose response curve shows us what happens to the response given a certain dose. So in this case, on our left axis, that's our y-axis, we have the height of the bag. That's what we're going to be measuring, is the height of the bag, aka the volume of CO2 in the bag, aka the cellular respiration of the yeast. And on the right, we have the concentration of salt in our bag in teaspoons. So that will be teaspoons per 2 tablespoons of water. So it's more just an amount than a concentration, but that's what we'll be comparing it to. I think next.
How's your yeast looking, Christian? I think you're muted. I'm definitely getting some air in my bags. Nice, nice. Yeah, so it's not crazy, but there's a good amount of carbon dioxide in there, I'd say.
Was I muted that whole time?
Yeah.
Okay. Like, even though I don't see a huge volume of gas created, I do see bubbles in there, indicating that carbon dioxide was indeed being made.
Yeah. Okay, so let's find out. Let's figure out how much air is in our bags. So the way that we're going to do this, I'm actually going to move my computer so you can see what I'm doing a little bit better. So I'll be right back. Okay, so what I'm going to do is I'm going to measure the height of my bags with my ruler that I have in my kit. And basically, I'm going to try to level out the top of this bag so that we can get a nice consistent reading. I kind of like to use something flat. It's a little easier than my hand. And I can prop that up and then just measure the height of your bags. So for me, this is my control bag. I'm putting my ruler up, and I want to make sure I'm looking at centimeters, not inches. So I would say this is at 1.5 centimeters. And I'm going to write that down on my worksheet in my table. Okay. Kristen, are you able to look at the chat?
Yes. They asked if we could stop sharing so that we could see the you measuring better.
Oh, absolutely. Yeah. Okay, thank you. Let me try this again. Okay, so I have my baggie. I'm either leveling it out with my hand or some kind of flat object to try to get the bag nice and even. And then I'm using the centimeters on my ruler to just measure the height of the bag, so the highest point of the bag. Okay, and then once you measure that, you just want to write it down in your table on the worksheet. And then you'll do that for each of your baggies. So we should have a height for the control, a height for the low dose, a height for the medium dose, and a height for the high dose. Okay, I'm going to move my computer back. And I'm going to take a second to measure my own bags so that we have some data to look at.
Emily, would you like to average our data together for our final data set?
Sure. Sorry, Christian. It'll be just a second.
That's okay. If you are at home and you are a little disappointed with the amount of air you're seeing in your bags and you want to wait even longer to see if they will grow larger and if you'll see a different or greater response later on, you are more than welcome to do that. And I imagine you'll get to see an even greater response that way.
That's a great point, Christian. I actually left my baggies out on the counter last time that I did this because I was going to clean them out and had a class to go to. And I came back and the control baggie was about ready to explode. It was completely full.
How long was that you let those sit there, Emily?
Oh, probably an hour and a half, I think
Okay.
is the length of that class.
So we might not want to let it go longer than an hour and a half so that our bags don't explode.
Yeah.
Okay.
Yeah, and if you are going to let your baggies sit out before cleaning them or throwing them away, maybe open them up so that they don't get too big. Okay, I have my data. I'm going to go to, oh, there's the table. Okay, so I entered my values into my data table. And then the next thing that we're going to do is we should have a graph and a grid on our worksheet. And we're going to plot.
Emily, it could be good for you to get out of the presentation again so we can see you hold up your worksheet. Or maybe not.
I'll switch it back once I actually get points on there. I think it might be clearer just on the PowerPoint since everything will be in reverse on the worksheet. So we want to plot each of our measurements on the graph. So on the far left of the graph, you have the 0 concentration, which is your control bag. So whatever your height was for the control bag, for me it was 2 centimeters, I'm going to put a point on the graph. And then you'll do the same for the 1/4 teaspoon of salt, which was your low dose. You'll plot whatever that was. For me, that was 1.5 centimeters. And you'll do the same thing for your 1/2 teaspoon and your 1 teaspoon dose. Then once you get those 4 points on your graph, you can draw a line through them. The shape of this graph here is a pretty common shape that we see with chemical dose response curves. So it's kind of like an S almost, like a backwards S. And so once you do that, we'll see if there's a trend in our plot, seeing if increasing the dose of salt either increases or decreases the height of our bag. So I will plot my points on my graph and then I will try to share those with you. Okay, I'm gonna stop sharing for a second. Okay, so this is sort of what my graph looks like. And you can see there's kind of some weird squiggles in the line, but for the most part, you can see that it's decreasing. So as the amount of salt I added to my baggies increased, the height of my baggie also decreased. So that's what we hypothesized. That's what we would hope to see. And have a graph, Christian.
What's that? Oh, no, I do not have a graph. I didn't have
That's
the
okay.
worksheet.
Is that kind of what your data looked like, or was yours higher?
Interestingly, my control baggie was less inflated than my 1/4 teaspoon of salt baggie. So I had first an increase and then a decrease.
That's very interesting.
Yeah. I would want to do more experiments to see if I saw a similar thing.
Definitely. Okay. So now that we have our graphs put together, let's talk about our results a little bit. So Christian and I compared a little bit, and you can compare your results to what we got. But let's talk a little bit about why we saw what we saw and how we can interpret our results. So the first thing is let's review what our independent and dependent variables were in this experiment. Do you remember from the very beginning what the independent variable for this experiment was? So the independent variable was the amount of salt that we were adding to the bags. And do you remember what the dependent variable is? This is the response, or what we're measuring. Yeah, so that would be the height of your baggie, or the amount of cellular respiration that is going on. And then we also talked about controls. We talked about sugar being a control, so each of your baggies have the same amount of sugar, and they should all be at the same temperature because you're using the same type of heating device. So I had all of mine on hand warmers, which should all be the same temperature. The location of the bags was the same. So what do those controls help us with in the experiment? If you remember, I think we talked about this. So all of those things ensure that when we're measuring our dependent variables, so the height of the baggies, those should be all comparable, because we've tried to keep everything the same except for the amount of salt that we're adding to the bags. Another thing that we had in this experiment was our control baggie. And for a lot of people, I would guess that this baggie had the most air in it, the most carbon dioxide. So what was the point of the control baggie, and what do you think that tells us? Just think about it for a second. So the control baggie gives us an idea of what we expect to see without adding any salt. So these should be healthy yeast that don't have any salt added to them. And so that should give us an idea of what we expect to see in normal yeast before we start adding salt to it. And that way, when we add salt to our bags, we can compare those heights to the control. And so in my case, all of my baggies that had salt in them, they had lower heights than the control baggie. So that tells me that all of the bags that I added salt to, those yeast were less healthy than the normal yeast that didn't have any salt. So it's a little hard if you're doing this on your own at home to compare with other people, but like I said, you can kind of compare to the results that Christian and I got. If you were to see different results in your house than somebody doing the experiment in another house, like Christian and I's results weren't exactly the same, what do you think might have caused those differences? Christian, what do you think are maybe some factors that are different between your house and my house?
Well, I used a hot water bowl to incubate my yeast, and you used hot pads. So that's one difference. Another thing is I wondered if maybe I didn't mix my yeast and sugar up enough or homogeneously, like in between all the bags, so that maybe the low bag had more access to sugar than the control bag.
Yeah, I was kind of wondering that too. I noticed I had some like pockets of yeast in the corners of my baggies that maybe didn't come in contact with the water or the sugar. That's a good point. I also, when I put mine on the hand warmers, I kind of scrunched up the bottom of the bags to get them to fit on there. And I was wondering if that might impact the yeast's ability to I don't know, grow? I'm not sure, but that's a big difference between our experiments, definitely. We might even have different size sandwich baggies, maybe, which would mean that we have different heights. So why would we replicate an experiment? This is something that's really, really important in science. So like Christian was saying, he did his experiment once and he got kind of a weird result where his low dose bag had a higher volume than his control bag. And that doesn't necessarily mean anything, but it's not quite what we expected. And in science, you want to do an experiment a lot of times to make sure that the results that you're getting are something that you can really make a conclusion from. Because if you only do something once, how do you know that that's what's going to happen every time? It could just be chance that that's the result that you got. So by doing the experiment over and over and over again, you can assure yourself that the results that you're getting are at least close to the truth. So how many times do you need to replicate an experiment? Is there a number? Is there a magic number, Christian?
I don't know what until you feel confident enough that things are doing the same thing, I guess.
Yeah, there's really no magic number. I mean, the more times, the better. Unfortunately, in science, we can be limited because experiments can be expensive and they can take a lot of resources. So if you were to do this experiment 100 times, that'd be a lot of yeast that you have to buy to do the experiment, for example. So the more times, the better, but definitely there are limitations to that. And finally, I'm going to pose a question. Why do you think the yeast produced less carbon dioxide when more salt was present? Why do you think that the salt was toxic to the yeast? So in my brief research, I think that the salt is not helping the yeast grow, it's harming their growth, because in organisms, you have a very fine balance of water and salt. It's very important that you maintain a steady concentration. And for us, our kidneys actually help with this a lot. But if that balance is thrown off, so if you have too much salt, or in the water case where we were saying you drank a bunch of water, the issue is that your salt is actually too low. Another way to think about this is after, say, you exercise really, really hard, you go for a run. Some people like to drink Gatorade, and Gatorade has electrolytes in it, or salts. So by adding salt to the yeast baggies, we're throwing off this balance of salt and water that's really important in organisms, and that can actually harm their growth. So that is all I have. Christian, do you have any other talking points?
Someone asked if we could redo the experiment, but with different products in our house. And I think the answer to that is that would be really exciting to do, as long as you have your parents' permission and they are safe products.
Yes, that's a great point. But yeah, I definitely encourage that if you wanted to try something else. And then you could even see which of those chemicals is more toxic. Is the salt more toxic than the soda pop that you have in your fridge? So feel free to experiment with that.